Introducing VitroGel, the most versatile synthetic hydrogel for 3D cell culture and beyond. A groundbreaking 3D cell culture hydrogel that is revolutionizing research and discovery. VitroGel is a pure synthetic xeno-free biofunctional hydrogel system designed to provide a seamless solution for your entire research journey from bench to bedside. GMP-grade VitroGel is available to support your research to go beyond the lab for biomedical applications. VitroGel is modified with multiple cellular functional ligands, which closely mimics the natural extracellular matrix or ECM environment, supporting various applications like 3D cell culture, lab automation, and in vivo injection. VitroGel's defined nature assures you batch-to-batch consistency that is free of undefined components that come with animal-based ECMs. VitroGel is user-friendly and remains in liquid form at room temperature. The gelation process is triggered when the hydrogel is mixed with cell culture medium rather than adjusting the temperature. This not only makes the operation relaxing and easy to control, but also eliminates the extra time needed for thawing and incubation, as well as the environmental burden and additional costs associated with dry ice required for shipping animal-based ECMs. Experience VitroGel's quick and easy 20-minute room temperature protocol free from any animal or human components. There are two versions of the VitroGel system designed to maximize flexibility and provide users with tailored options. Ready-to-use VitroGel hydrogels are your go-to solution for a fast, user-friendly setup. With optimal formulations across cell types and applications, it saves you time and effort while delivering reliable results every time. For those who need more customization, tunable VitroGel high-concentration hydrogels are game changers. These tunable hydrogels give you the flexibility to adjust the hydrogel's concentration, empowering you to create an optimized environment for cell growth based on your research needs. Each of the 6 tunable VitroGel hydrogels can be mixed and matched with each other to create a customized multifunctional hydrogel for different applications. By using VitroGel, you're equipped to push boundaries for advanced, customizable research. Whether you choose the ready-to-use or tunable high-concentration version of VitroGel, its unique injectable properties set it apart. The VitroGel system seamlessly transitions from a gel to a solution during injection and rapidly recovers to form a hydrogel afterward. This exceptional shear thinning and rapid recovery rheological behavior makes VitroGel an ideal solution for applications such as injection, bioprinting, droplet formation, and lab automation. From in vitro research to in vivo applications, VitroGel adapts to your needs. From cancer studies, organoid co-culture, and stem cell applications to patient-derived and cell-derived xenograft, cell injection and tissue regeneration, VitroGel offers unmatched flexibility and reliability, supporting your success in your scientific research. 3D cell culture doesn't stop with the VitroGel hydrogels. The 𝗀𝗶𝘁 provides an end-to-end solution, starting with the VitroGel Organoid Recovery Solution, an enzyme-free media that allows you to quickly and easily harvest cells in less than 20 minutes, all while preserving high cell viability. This efficient, gentle method keeps your cells healthy and intact, making it ideal for downstream applications and high-quality results. Beyond cell recovery, our comprehensive product line also includes the premium CytoGrow growth factors, Vitro Prime Cultureware, rocket cell culture media to downstream platform assays like our Cyto3D Live Dead assay kit. Cited in numerous notable studies worldwide, VitroGel has been instrumental in advancing breakthroughs in precision medicine, cell therapy, and advanced biomanufacturing. Its unparalleled versatility and reliability make VitroGel the top choice for innovative and transformative biomedical research. Start with the end in mind. The future of 3D cell culture begins with VitroGel.